Morning guys, welcome to another Biology with Miss Cherry. Today we're moving on to statistics and looking at how they link in with genetics. So to begin with, have a look at this starter question. Think back to what you know about statistics, think about what you know about statistical tests and significance, and then when you're ready, press play and we'll get started in the lesson. We'll come back to answering these questions towards the end. So today's lesson is all about the chi-squared test. You can call it the chi-squared test, but I don't think that's the right way to say it. So you pronounce it chi-squared, and the symbol for the chi-squared test is a big X, the symbol chi in the Greek alphabet, and then the squared like you'd normally put. Now we're going to look at how to do the chi-squared test. We did the required practical about wood lice, so we already know how to do it, but we'll recap that, and then we'll see how it links into genetics. So let's start here. Imagine we roll a dice 20 times. How many times would you expect it to land on 1? Okay, just due to the randomness of life, the chance of life, how many times out of 20 would you expect it to land on 1? Now realistically, you'd expect to roll a 1 a sixth of the time. Okay, there's 6 sides on a die, and every side has got an equal chance of being rolled each time, and this is important. The rolling of dice is completely random. However, what happens, or what does it mean, if we didn't actually roll 5 ones out of those 20 times? What could it mean? What reasons could there be for that difference? So you can pause now, have a think, why is that variation, that difference in what we expect to happen and what actually happens, the observed values for that variable. Now if the dice, not rice, rolls a 1 more than 5 times, could be completely random, could be to do with just the way that life's going that day, but there could also be another reason, and that's the whole idea about this chi-squared test. Okay, there could be another reason other than chance causing that difference. So something like maybe I've stuck a bit of blue tack on the 1, for example, and it gives it a little bit more weight. And again, this is the same if we roll a 1 less than 5 times, so there may be another reason as to why that 1 is less favoured in the random rolling of a dice. Now the chi-squared test is used to look at the magnitude or the size of the difference between the observed experimental data and the expected theoretical result. Okay, and this is generally, we say that the chi-square test is there to measure the goodness of fit. How well does what actually happens link to what you expect to see? And if it's not very well linked, then we can think of reasons why later on about that. Before we have a look at an example though, we need to recognise that there are some caveats to using the chi-squared test. There are some rules that we need to follow, and there are some principles that we need to adhere to ensure that our results are going to be reliable and accurate. So the first one is that you need to have a relatively large sample size. It's no good looking at observed and expected results if you've got maybe 5 organisms. It's not very helpful. So generally we say we need at least 20 individuals, maybe at least 20 people in a drugs trial, maybe at least 20 organisms in a distribution study. And those organisms have to fall into discrete categories. So they've got to be a categoric set of data. So they can be maybe like blue and short aliens, black and tall aliens, blue and tall aliens, and black and short aliens. But they're not saying, oh, these have got a height of 162 centimetres. Okay, we can only use raw data with it. So we can't use process data, not percentages, not ratios or anything like that. So this is straight from an observation perspective. And finally, we're using it to compare that observed data with the expected data. So remember, it's the experimental versus the theoretical. And this is how it links into genetic crosses. We'll take a look at that in a little bit more detail in a bit. Now, the final thing that we need to be really confident in doing is writing a null hypothesis, because this is the bit that generally starts off a really good 6-mark answer for the chi-squared test exam questions. So a null hypothesis is where you say that there is no significant difference between the observed and expected ratios of something due to something other than chance. Okay, so as a standard answer for this, so the null hypothesis, which you can write as H0, there is no significant difference between the observed and expected ratios of, and then you put your variable in there, so maybe we're talking about blue tall aliens due to anything other than chance. Okay, and this implies that it's just the randomness of life that is causing that change. And if we, at the end of the chi-squared test, either accept or reject that null hypothesis, then we can suggest reasons or other reasons why there might be a difference between those two ratios. Okay, so let's get started with an example. We'll work our way through it, and then you guys can have a go at one afterwards. So before we go anywhere with our example, we're looking at the equation itself. So it looks a little bit like this. Not necessarily going to be given it in an exam situation. Okay, so the chi-squared value is there. And then we've got sigma, which means the sum of, it's a big Greek letter, and it's the sum of the observed minus the expected frequencies squared divided by the expected frequency. So the easiest way to do this is with a table, and that's how I'm going to show you how to do it and provide you with on the crib sheet that I've attached to OneNote. And hopefully that helps you lay it out in a nice standard format. In the exam as well, sometimes they give a table ready for you to fill out as well. Now there are a few other details that we need to be aware of before we get into actually having a look at those answers, or that example anyway. And that's about things like the degrees of freedom, which in an exam situation you can just abbreviate to DF. And that's basically a measure of how many groups that you've got. Okay, but the way you calculate it is you have to take one away from the number of categories. So if we think back to that aliens example, we had 4 different types of alien, so my degrees of freedom would be 4 minus 1 or 3. Hopefully in the exam they won't give you too many categories of phenotypes or something like that. It would be a bit mean if they did. The next one is looking at how your chi-squared values relate to the critical value. And unless you can remember back to when we did that Woodlouse practical, the maggot practical, this isn't really going to make much sense. So we're going to leave these bits for now, and we'll come back to it when we look at the example at the end. Okay? So this is the example that we're going to work through. It's exactly the same one as we did with the choice chamber practical from module 6. So imagine I've got 5 woodlice dropped in a choice chamber, and they've got a moist environment on one side and a dry environment on the other. And I'm going to leave my timer 4 minutes exactly. Okay, so I'm controlling that variable. And after that time, I'm going to have a look at how my woodlice have distributed themselves between those two areas. The first thing I do is I start with my null hypothesis. So in an exam, you can just write HO, or you can write null hypothesis, and you can say any differences in observed and expected distribution of woodlice between the moist and dry areas are due to chance alone, or there is no significant difference between the observed and expected ratios of woodlice in the moist and dry areas due to anything other than chance. Okay, so remember when we said that sort of basic standalone statement, the variable that you're putting in here is what you're testing for. So you're looking at the distribution of woodlice between moist and dry areas. So that's the first thing you're going to do. Null hypothesis, write it down. Single standard statement, get started with that. Then you've got to figure out what your expected distribution of woodlice are for each area. So in this situation, if there's no extraneous variables or factors that are affecting the woodlice movement, then you should expect them to be evenly distributed. And of my 5 woodlice, I would expect 2.5 in the moist area and 2.5 in the dry. We're not going to think about the fact that there's half a woodlouse in either area. We're just going to work with it. chi-squared test is based on this 1-to-1 moist to dry wood lice quantity ratio. And then I have to do my investigation. So my expected ratio is 1-to-1 moist to dry. But in actual fact, I saw 5 wood lice in the damp area and 0 in the dry area. Okay, and that's what I'm going to use to produce my table. And it looks a little bit something like this. So I've got my two variables, my two areas on the left-hand side of the table, and I'm going to put my observed first and expected afterwards, because that's going to help the calculation make a little bit more sense in my head. Because if you remember in the bracket, as you can see on the left-hand side, it's observed minus expected, not expected minus observed. So if you just put those in the right order straight away, makes it a little bit easier for next time. And then I'm going to finish my table. So I've got my observed and expected, and then I'm going to start doing the calculations that are in my formula. Okay, observed minus expected squared, and then finally at the very far side, observed minus expected squared over expected. And you see, taking each of those columns in turn makes the whole table a little bit easier to flow and work through. Okay, but now I'm going to be focusing on this square, this blank down here. Because remember, our chi-squared value is the sum of the observed minus expected squared over the expected. So this is where our chi-squared value lives. And to figure out my chi-squared value, as I've said, I'm just going to work out what goes in that box. So I'm going to add the 2.5 and the 2.5 together. So my chi-squared value for this investigation is 5. And remember, I need to figure out my degrees of freedom as well. So I've got 2 different options, or 2 different groupings of where the organisms can be. So my degrees of freedom is 2 minus 1, and therefore my degrees of freedom is 1. Now, this is where the degrees of freedom, and we go through how to read this table. Okay, this is how we find out whether there is or isn't significance. So we need to use our chi-squared value, which was 5, and we've got a degrees of freedom value of 1. So degrees of freedom, you can see on the left and the right hand side of the table, the far left and the far right, they've put them at either side just in case you're looking at one side of the table or the other. And what we're doing is we are highlighting this row. This is where we know we're looking. And unless it says otherwise, you are always focusing on the column that says P equals 0.05. That's to do with the percentage chance. So that means that there's a 5% chance that your results are due to chance. Okay, and then we're going to take a look at how to read these tables in a second. And then what we're doing is we're figuring out whether our result is or isn't significant based on these 2 red and green rules that I've got at the bottom. Okay, so I know that I'm looking at 1 degree of freedom. I'm looking at that row across the top that's got 1. I know that I'm looking at the 0.05 critical value level, the 5% value. And then we've got these 2 rules, as I've said, down the bottom. So if your calculated chi-square value is to the left of that 0.05 value, so it's between 0.02 over here and 3.84 really over here, then you have to accept the null hypothesis. There is no significant difference. Okay? Any difference is due to chance. It's completely random. Conversely, if it's to the right of the value, so it's bigger than the critical value that you can see in the table, then you reject the null hypothesis. Okay, and there's probably another reason, something other than chance, causing that difference. So pause now, maybe take a photo of this, make a note of how to use the critical value table before we look at our results and see where they sit. Actually, while you're doing that, decide how you would summarise whether our value at 1 degree of freedom is or isn't significant. Okay, pause and play again when you're ready. So what you should have got is that the critical value is 3.84, 3.85-ish, and our calculated chi-squared value is 5. And as 5 is bigger than 3.85, so it's to the right, and we reject the null hypothesis, there is a significant difference. And you have to have a little summary statement. Yeah, it's a good thing to have that little rounding off. You recognise that I'm rejecting the null hypothesis, there is significant difference, and there is something other than chance causing it. A good practice to start embedding in how you do these questions is to maybe mention or suggest a reason as to why there might be that difference in your summary statement. So you could say a taxis. If we're talking about plants, you might want to say a tropism, for example. Okay, now let's have a look at how it works with genetic cross. So here we've got a cross, a monohybrid cross, so we know we're looking at one gene only, involves 2 heterozygous purple flowers. So heterozygous, we know we're going to have one dom and one recessive allele. The F1 generation resulted in 105 purple flowers and 45 white flowers. This is our observed value. Okay, this is the experimental stuff. Okay, so the first thing we do, we start off with our null hypothesis. So that null hypothesis needs to come first. Then we need to figure out what we would expect to see of each phenotype. We're then going to go through the process of doing our chi-squared table, and then we're going to compare it to the critical values. So we'll take each of those in turn now. So, null hypothesis. Any differences in the observed numbers of purple and white phenotypes, okay, so remember that's our variable, from the expected numbers are due to chance. And again, there is another way you could write it, so there is no significant difference between observed and expected numbers of purple and the white phenotypes due to anything other than chance. Okay, so we've got that horrible scruffy sentence, but you get my drift. And then what we need to do is figure out what our expected ratios are. Because if you think back to the previous question, they gave us the values of what the offspring actually are. So from the question, it told me that I'm looking at a monogenic heterozygous parent being crossed with another monogenic heterozygous parent. So I'm going to smash those p's into a Punnett square. So big P, little p, big P, little p. And again, I'm not following my own rules. I've chosen p's for this one. It was in the book. But p's are preferred not to use because, as you can see, it's really difficult to tell the difference between a capital and a lowercase p. And your ratio should be 3 purple to 1 white. Okay, so our purple phenotypes I'll highlight in, or our purple genotypes I will highlight in blue. So any dominant P allele, we know that is going to be the purple flower. So the phenotypic ratio is 3 to 1. Now we need to figure out how many offspring are going to be purple in that 3 to 1 ratio. So we're then going to have a look at how to calculate the number of offspring that you expect to see from that ratio. So in the previous question, or in the stem of the question anyway, it told us that we have got 105 purple flowers and 45 white flowers. And that gives us a total offspring number of 150. Okay, so that's the number that we're working with. Now from our ratio, the 3 to 1 purple to white ratio, we know that 3/4 of that 150 need to be purple. Okay, so 3/4 times 150 gives us 112.5 offspring. And likewise, for the white flowers, 1/4 of them are going to be white, and that gives us 37.5 flowers. Remember, we're not too worried about this half value just yet. Okay? In fact, we're not worried about it at all. It's chill, we can work with it. So let's have a look at that in the table, and we'll see what our chi-squared value is. Now here you can see that we've got the phenotype table. I haven't done the whole thing this time. I've just done it as an equation down the bottom. So we've got our observed minus expected squared divided by the expected. And that gives us a chi-square value for white as 1.5 and a chi-square value for purple as 0.5, giving us an overall chi-squared value of 2. Remember, we've got our chi-square value of 2, but we still need our degrees of freedom. There's 2 different options for that phenotype, either purple or white. So we've got 1 degree of freedom in this investigation. And the final thing we're going to do is compare it to our critical value table. Remember, we're always looking at that 0.05 column and 1 degree of freedom on the row. Now our chi-squared value this time, our chi-squared value of 2, is definitely to the left. Okay, it's lower than that 3.84. And therefore, we accept our null hypothesis, and there is no significant difference between the observed and expected ratios. It's not significant statistically. Okay, and the differences are just going to be due to chance alone. Now, this is a chance for you guys to do something by yourselves. So a corn has 2 genes controlling the colour and texture of seeds, and we've got big P's and little p's for purple and yellow colour, and then big R for round and little r for wrinkled. A study on 2 heterozygotes for both colour and texture were cross-fertilised, and the cross of the F1 generation was analysed. Now this is important because there's a lot of faff, a lot of information that is important but isn't. So let's pick out the key details from this question. We know that we're looking at the key for colour and for texture, and we know that we're looking at 2 heterozygotes being crossed, and they are analysing the F1 generation. Okay, so cross-fertilised means that they're just breeding them together. So in this question, you need to complete the table, and what statistical conclusions can you make about the genes from the results. So I've given you the observed, you need to figure out the expected, complete the table, calculate your chi-squared value, and then use the statistical table on the next slide to find out where your chi-squared value sits and give me a summary statement. It's 12 marks, so it should take you about 15 minutes to do. Okay, so pause now. And when you're ready to go onto the table of critical values, the ones with the P's and the degrees of freedom and stuff, then play, and I'll just skim over it and you're going to have to pause it again. All right, so pause now, 15 minutes, play when you need the table. So here's the table, pause again, find out where your chi-squared value is, and write your summary statement. So here we've got the answers for the table. I've skipped out all of the observed minus expected squared, observed minus expected squared over expected, and just given you the expected numbers that you should have got from a Punnett square. Okay, so it should have been a 4x4 Punnett square, because we've got 2 heterozygotes. So we had P, P, R, R times P, P, R, R. You've got your 1, 3, 1, 4, 2, 3, 2, 4 things going on. So we've got big P, big R, big P, little p, big R, and then little p, little r. Stick those in your Punnett square, and you should have got a 9, 3, 3, 1 ratio. And I've done these to the nearest offspring, so the totals might be ever so slightly off. You could have done them in decimals as well, but they should be roughly the same as what I've got. Now the chi-squared for each of the phenotypes is on the right hand side, and that gives us an overall chi-squared value of 1.8. Now for this table, each row that you've got correct is a mark. Okay, so we've got 5 available here, so 1, 2, 3, 4, 5. If you're not sure how close your answer is to being acceptable because you've used decimals and things like that, hit me up on chat and we will see where you stand on that. But that's what we're carrying forward. So I'm taking forward this 1.8. My degrees of freedom is the next mark. So that's 4 minus 1. Degrees of freedom is 3. Calculated chi-square valued was 1.8. That's what we got. The critical value for 3 degrees of freedom at 0.05 is 7.81. and that 7.81 is bigger than my 1.8. So my chi-squared value is somewhere over here. Okay, so there's a few marks left for these final summary statements, and this is why they're so important to do in the exam. Firstly, we've got to establish what the relationship is between that critical value and my chi-squared. So chi-squared, and that's the crit. So that's your first mark. Therefore, there is no significant difference between the observed and expected. And therefore, again, remember we need to suggest a reason why there is that difference or what the relationship between them is. We can say that the genes are not linked. Now, you could have given another reason due to chance, and again, you might want to just talk to me on chat and find out whether your answer would be appropriate or not for that point. Now, the final thing you're going to do, hopefully if you got all of those right, is put the summary questions from page 181 in the Oxford Blue Book, so the Year 2 book, on our OneNote. Have a go at those and then let me know when you need the answers and I'll put them up there as well. Okay? I hope you found this useful. Chi-squared test is a little bit tricky, but I will see you again next time for something that will probably be just as mathsy. I think it's the Hardy-Weinberg equation, so that'll be fun. Bye-bye!